Hey, how are you?
Good. How are you? Alfred.
Good.
Can you see me?
Yep. Yeah.
Cool.
Thanks for joining.
Oh, thank you. Oh, I need to change my name. Sorry. That was the name of my son.
Okay.
Okay. How do I do that?
I can do it for you.
No worries. I got it. Yeah. So we just had a parent-teacher meeting. So I have to log in with my son's credentials. And I forgot to change it back.
Do you want to test out your slide?
Of course, sure. And am I the first one or the second one?
That's a good question. I thought you were the first one, but then I looked on my. Well, let's see. Would you like to be the first or the second one?
It doesn't really matter.
Okay. Yeah, sure. Yeah, usually I list it in the. Yeah, that looks great.
Okay.
Yeah. Normally I list it on the website on the flyer as
go from
order,
the left to right, right? Yeah.
Yeah, but this time it's sort of like it's different. So yes.
Okay.
Yes, you can go first.
I'm sorry. Okay.
Yes. You go first.
Okay.
How's everything in Singapore?
It's we've been up and down. We never really go fully out and never really be like China, clear, have a zero tolerance policy kind of thing. So it's been difficult. Yeah, that's why my lab has been so much delayed and can't really have everything properly started. Hello.
Nice to meet you.
Hi, Istvan.
Hi, Aaron. Nice to meet you. Hi, Alfred. Nice to meet you.
Nice to meet you.
Istvan, can you give your slides a test?
Yes.
Thanks for joining us.
Can you see that?
Yes.
Yep.
And can you share do the screen, like the slideshow, just to make sure that looks good.
This one. Can you see my mouse, my cursor?
No. It looks like it's not. We don't see the slideshow view.
You don't see the slideshow view?
No, can't see that.
Wait a sec. I escape. This is what you need.
Oh, it's moving now.
No, but go to the the
slideshow.
Like yeah.
Wait a sec. Is it visible now?
No, it's still no. Do the one where you have like the where you're giving where you're giving a presentation.
Yeah, I think this will work.
Can you see
like this one, this button? Yeah.
Okay, I try something. Wait a sec. Okay.
Sure.
Can you see and hear me?
Yes.
Okay, so do you see my screen?
No, you have to share it.
I need to share again. Okay.
Okay.
Can you see my screen now?
Yes.
Yes,
Good.
Alfred, you can see. Aaron?
Yes, yes.
Did you
can see
get?
it. Yeah.
Right.
Yeah, but now, now click that button. Good.
Okay, so now I do the slideshow. Can you see the title and your name?
Sure.
Yeah, no, there you go. That's good.
Can you
looks
see?
good.
Looks good.
Looks good. Looks great.
Can you see my mouse, the pointer?
Yes.
Yeah.
Yep. Perfect.
Okay, then everything is fine. So
Great.
This is really amazing what you are organizing. I checked the previous lectures, some of them even listened, and it's really amazing. Congratulations and thanks a lot.
Oh, thanks, but it's really thanks to everyone who's
totally agree. I just very curious, how did this thing started between you and Zilong?
I did all of the works.
Is it because of COVID or it's before COVID?
No, it's COVID. It's just really, yeah, shortly after COVID.
I see. That's really good. Yeah.
Yeah, it's because normally the 4 p.m. seminar in Stanford cannot be going on. So Aaron is so bored. So talk to me about starting. So whenever the regular 4 p.m. seminar is going on again, I guess like the 5 p.m. this we should maybe find another time something. Yeah.
I see. Just wondering, this is only for neuro or any? Are you guys aware of any other disciplines that are doing something similar?
Right. So after we invited a couple of like neuro-immuno guy, they thought, wow, it's great. Then it's starting to the immuno Zoom.
Oh, I see.
There's a brain Zoom. So after neuro Zoom is several, so the
But this is original.
Oh, of course, yeah.
Fantastic.
The brain zone and the immuno zone after.
Brain zone. Okay.
Yes.
Yeah,
I
that's
think we're the only ones that have like hoodies and things associated with it.
Hoodie.
It is on sale. Where am I going to get it?
You can't buy it. You can't buy it.
And Aaron, I have a question for you. Previously, I saw the last couple of Zoom talks that your background was actually a photo that I'm very familiar with. Was that from Hangzhou?
Yes. Yeah. I'm in China now. I'm in China.
Oh, you are?
Yeah, I'm in Shanghai. Last week I was in Hangzhou.
Oh, no wonder. Yeah, because I was born in Hangzhou. So
yeah.
In fact, the same school that Hailan Hu attended. She's my senior. Yeah, great. I couldn't go back for the last 3 years, so it was really nice to see at least you use that photo as a virtual background.
I went after her talk, or I went on that trip. We went hiking all through like the West Lake area, Hailan and me and a couple others.
Oh, fantastic!
Yeah, it's really good there.
Cool.
And then like the day after I left Zhejiang University, there was a shutdown because there was like one COVID case. So I was like luckily escaped.
Yeah. I was going to say, many of my friends were still couldn't go home that 2 days or something.
Yeah, the lockdowns here are serious. They lock you down. You can't leave.
Oh yeah, more than 60,000 people cannot go home, and yeah, they have to clear you.
In the lab, one kind of fly that start to cooking use the hot pot use.
Oh no, really?
Seriously.
Yeah.
Li Ming, the friend Li Ming Wang, so he got some rice and the cooked canned food. So the students started cooking with that.
Wow, and it's pretty cold because it's winter time now. So
it's not that cold. Yeah.
So Aaron knows that I just got COVID. Actually, this is my last day in isolation. From tomorrow, officially, I can go back to work. But I was at the Gordon conference 2 weeks ago, and although it was a fully vaccinated crowd there, unfortunately, a few people got it, or a few dozen people got it. So it is still around.
Were you? Hopefully things were mild.
Yes. It was okay, so I don't complain. I have 3 children and they didn't vent to them, and that was the most important thing. So
Oh, so even after Golden Conference, come back, you need to.
Yeah, and then some colleagues told me from the building that they are already positive, so I should test myself. So I went and did a kind of free test for myself, and oops, detected. So then I stayed home. So I hope we could stop the chain.
Did you lose smell or taste or anything? Or
yeah, a little bit smell, yes. Yes.
So for my family, pretty much everybody has got COVID. I have 2 of my boys and our helper, and one by one, we couldn't even, the government say self-quarantine at home, but we couldn't, and like isolation, but we ran out of rooms pretty much very quickly. So
I am in this room for 10 days. I just don't show you around. I'm sorry. This is actually a lecture hall and workplace and bed and everything is here. So. Oh, hi, Ming Hong. Nice to meet you.
Nice meeting you.
Nice to meet you. Finally on internet.
Oh, hi, Kam.
Hey, Alfred. Good to see you.
Hi. Good to see you. Do you still remember me?
Yes. Of course.
All right, Aaron, let's get started.
Okay, good. It's long. So we decided that Alfred will go first, but first I'd like to welcome everyone to another Neuro Zoom week, and like to advertise next week's talks. So we went through a lot of effort to get Rao Yi to talk. He's coming to talk, and he has a really exciting news story about sleep, both controlling and indicating sleep. He discovered it down to a single phosphosite. And then we have a wonderful talk from Professor Viviana Gradinaro from Caltech. She's a legend in her own right, and we'll talk about new technologies and also exciting studies about Parkinson's disease and alpha-synuclein. And then keep tuning in. We have many more great speakers for you coming up in the coming weeks and months. And these are all leaders in the field. You won't want to miss Hu Zhao's talk, amazing new clearing technology that's going to revolutionize what we do. And of course, Hong Kui, others. And then we'd love to hear your talk. So please let me or Zilong know if you'd like to share your new results. Okay, so now Zilong, you can please introduce Alfred.
Great. So it's my great pleasure to introduce Alfred Sun, who is a good friend of mine over the years. Actually. So I met Alfred as like 10 years ago, right? So he just finished his postdoc. So Alfred has grown up in Hangzhou, a beautiful city nearby Shanghai, where all of you should go when you visit China. So after middle school, high school, Alfred went to US to has his bachelor degree in Duke University. So after a bachelor degree, he went to Stanford to have a PhD degree with Jerry Crabtree, where he started to learn how to culture stem cells and iPS cells. He started to work on stem cells ever since. So after post PhD, he went to Singapore to start his postdoc. I believe this, is with Dr. Zhu with the iPS cells, derived organoids. Has done a series of amazing work of the years. He just started his own lab in the National University of Singapore with joint program with Duke University to start his own program. And today he will talk about the use organoid to study Parkinson like the disease. To title is the Lewy body like inclusion in human midbrain organoids carrying PD like the mutations. Welcome, Alfred.
Thank you. Can you guys see my screen now?
Yes, looks good.
Great. Indeed, thanks, Zilong.
Sorry. Yeah, got it.
Oh, okay. Yeah, thank you, Zilong, for a very wonderful introduction. Indeed, I think it's about 10 years. Except that time I was just finished my grad school. And so today my talk is going to be on the Parkinson's disease, and we're talking about new Lewy body-like inclusions that can be found in human midbrain organoids carrying certain combinations of Parkinson-related mutations. So this, I hope everybody just had a very good Thanksgiving, and this slide is by no means to upset anyone. but I just want to, purely as a way of introduction, I would like to share one of my favorite quotes that all models are wrong, but some useful. And I would like to first focus on the useful part of this part, and to share with you particularly the last model shown here. Can you guys see my mouse? Or can I?
Yes, we can see.
Oh, okay. Yeah. So these, for those of you who doesn't know what these are 3D aggregates of human cells that are derived most likely from, most probably from human pluripotent stem cell, that is ES cells and iPS cells included. So this is sort of relatively newer models that people use to understand human relevance. Okay, so for Parkinson's disease, that is really a very common neurodegenerative disease. I just would like to highlight two of the pathological hallmarks of Parkinson's. The first one is actually the loss of pigmented dopaminergic neurons in the substantia nigra of the midbrain. So the keyword is actually those dopaminergic neurons are selectively dying, and they are actually a little bit black or brownish because they have they contain the pigment known as neuromelanin, right? So as you can see here. Now that's a very interesting feature, specific or rather to the primate and above, and particularly enriched in the human in an age-dependent manner, which means that in the rodents, in mice and rats, actually their midbrain are white because they do not, their dopaminergic neurons of the midbrain do not contain neuromelanin, right? Now the other feature of these Parkinson's hallmark is actually you have cell death, but you look at some of the remaining neurons, and you will see that they contain abnormal protein aggregates, in this case known as Lewy bodies and Lewy neurites, in which a major constituent is actually alpha-synuclein. So those aggregates are quite prominent features of the constituting the so-called Lewy pathology. Now, in this case, various animal models have been used to study and uncover a tremendous amount of insight about Parkinson's disease. But the one thing that is still lacking is that none of those models actually fully recapitulate the progressive sort of degenerative features of human Parkinson's disease. Even these two major hallmarks are actually quite challenging to recapitulate simultaneously in any single models, be it transgenic mice or genetic mice models carrying specific human Parkinson familial PD mutations, or using human cells in 2D cultures, the ones that we are more familiar with. So that posited when I started my postdoc in Singapore, and because my mentor is actually a clinician expert in movement order, particularly specializing in Parkinson's. So we thought that one way to look at this is perhaps have a newer model. That time we were inspired by some of the great works by Sai and Lancaster using making the brain organoid model. So we hypothesized that maybe a 3D organoid model of the human brain would better recapitulate human pathology and or pathophysiology in the case of Parkinson's. So because this has been published a couple of years ago now, so I'm going to make a very quick recap of the work. So essentially we're leveraging on the knowledge we learned that the mesocephalic dopaminergic neuron, particularly the ventral midbrain dopamine, are derived from the foreplate. So and take advantage of that, and they're right there in the middle. So by optimizing the dose of the Wnt and sonic hedgehog, as well as later FGF8 signaling pathway, we are able to make organoid, a 3D aggregate of the starting from human ES cells or iPS cells. Now, to characterize that, we use immunofluorescence as well as RT-PCR. So this is just really to show you that they contain multiple neural epithelials in a laminated way. pretty much mimicking the developing mouse midbrain intimate structure. And they also express prominently the floor plate markers, the classical floor plate markers, FOXC2 and NS1A co-expression of these two, together with OGX2. And later, you will see that in the outside, in the differentiated zoom, that NER1, a very important orphan nuclear receptor, is highly upregulated, thus showing these authentic midbrain dopaminergic neurons. And the bottom panel is just to show you that most of the key markers for the dopaminergic neurons are upregulated in a time-dependent manner. Next, we look at the other aspects of this, and we show that the human midbrain organoids actually resembles human midbrain in a way that, and it's different from the other brains, say the hindbrain or forebrain. And we realized quite interestingly that it seems to lies closer, which means it seems to be resemble more of the midbrain as compared to the 2D cultured dopaminergic neurons. So in this case, we are using the CRIX protocol developed by Lawrence Duder's group a couple of years ago. If you look at the organoids in a little bit longer time, you will see that they also express the dopamine transporter, the DAT, which is a very important marker for the function of dopaminergic neurons, as well as some other marks that marks the subtype of dopaminergic neurons in the midbrain. So there are A9 and A10, and people think that the GERK2 and Cal binding preferentially label these two groups respectively. And we have evidence showing that actually in our midbrain organoids, both types seem to be present. So both A9 and A10 are there. And we also worked in collaboration with Sean Jay's group to look at some of the electrical properties of these neurons. And indeed, we see that, for example, the sag voltage is quite prominently present, as well as the rhythmic discharge of these cells, again, reminiscent of the functional dopaminergic neurons in the midbrain. Okay, so this is what you get after 1 month of culture. We have millimeter scales of dopaminergic neurons, organoids that contain dopaminergic neurons. But really the interesting part, if you culture them a little bit longer, now this becomes like this. Okay, so I was very happy when I see this, and because we know that we could be hitting something quite important. That is, as I mentioned earlier, only the human midbrain dopaminergic neurons are black, and because of the neuromelanin. So we went down to further test, and sure enough, these are not contaminations, and because of the series of experiments, some of these are put in here. So when we look at the H&E stainings, they look similar as compared to the postmodern human and midbrain tissues. They're similar. We also look at we also extract actually the neuromelanin granules from the organoids and compare structurally to those isolated from the human substantial nigra. And again, they are very close to each other. Now, as I mentioned earlier, that the neuromelanin is actually a human-specific feature, right? So to directly actually look at that, we actually cultured organoids side by side, also with mouse ESL derived. So we made a mouse, a midbrain organoid, if you will, and we will find out that despite the fact that we have equal amount of dopaminergic, roughly dopaminergic neuron as marked by the marker TH, these organoids are never black. So again, this just highlights that this 3D model, for whatever reason, is able to precipitate this neuromelanin feature better. Okay, then to move on, after showing this model, we are wondering that if this, midbrain-like organoids in any ways can inform about PD pathogenesis. Now we know, again, as a reminder, there's two major hallmarks: dopamine neuron degeneration, as well as abnormal protein aggregation. So the way that we decided to do that is actually using a genetic approach to model those PD-linked genes using this organoid. We focus initially on several. I'm just highlighting two here. The first one is actually a GBA. So this includes an lysosomal enzyme known as GCase. We choose that because this is actually probably not the most strong PD-linked gene you can think of. The second one is alpha-synuclein is. However, it is actually the #1 risk locus, right? So we decided to include that in our studies. And of course, we also used alpha-synuclein overexpression mediated by lentiviruses to sort of mimic the case of alpha-synuclein triplication, which causes familiar cases of Parkinson. So this slide is showing you some of the characterization work to show that our approach worked. In the case of the knockout of GBA, we see that the heterozygotes have about half of the enzymatic activities. When the knockout is completely gone, and indeed we see that the substrate of the enzyme is accumulating, so GSLs accumulating in the organoids, but the products are actually decreased, as shown here. So in the case of alpha-synuclein overexpression, we put in a tag there. In the N-terminus, this is HA tag, so allow us to distinguish between the endogenous alpha-synuclein as well as the exogenous expressed alpha-synuclein, given by the upward shift in molecular weight. Okay. So we culture them as what as I briefly described before, and we noticed that the organoid actually can be formed very nicely. There's no problem in terms of formation of anything in these organoids with the mutations. However, we noticed that at day 60, the organoids are slightly smaller, as shown here. So they actually, the difference is not big, but it is quite reproducible. Next, we went on to see that if there is an early lineage commitment defect in those organoids that carry the mutations, and our evidence suggests that no, here's the data. Because when we look at the proliferation markers or any of those floor plate defining markers, OTS-FOXA2, for example, and when we quantify those, there's no statistically significant differences in any of those mutations that were tested. This suggests to us that the early lineage commitment seems to be by and large all right in those organoids. However, so what causes these slightly smaller organoids at a later time point, day 60 and later? We realize there is actually a specific reduction in dopamine neurons. So here is a quantification shown here. We see that both mutations result in a sort of partial decrease in the percentage of dopaminergic neurons as defined by TH. Now, the organoid does not only contain dopaminergic neurons, it also contains other cell types, progenitors, other type of neurons, astroglia. But in particular, we saw evidence that there are GABAergic neurons in the organoids. So when we quantify by GABA staining, actually the GABA population in the organoid is largely spared. So it looks like this is a fairly specific reduction in the dopaminergic neurons population. We then went in to use immunohistochemistry to look at the alpha-synuclein aggregation specifically. We saw that actually the mutant organoids exhibits certain aggregation phenotypes in terms of alpha-synuclein staining, and it is quantified here. And in all cases, it looks like the alpha-synuclein overexpression is able to produce more aggregates, perhaps not surprisingly, but the GBA knockout alone is also able to bring out certain aggregations in terms of alpha-synuclein. This is consistent with a lot of works from Joseph Mazuri's and Dimitri Kranik's lab, so suggesting there's a crosstalk between GBA and alpha-synuclein. We then look at this aggregation a little bit differently. This is a biochemical fractionation experiment when we look at the detergent soluble and versus detergent resistant alpha-synucleins. We see that again in the G means GBA knockout, the S means alpha-synuclein overexpression. That there is both mutations seem to give rise to more detergent resistant and become insoluble alpha-synuclein, as shown here. interestingly, symphyline, another one that had a protein that is implicated in aggregation, particularly in Lewy body formation, is also upregulated in the insoluble fraction of the mutant organoids. We then quantified the amount of viable cells, as well as so looking at essentially using flow cytometry to quantify the percentage of dopaminergic neurons that are undergoing apoptosis by looking at the CC tree, Cleve cascade tree. So we found that alpha-synuclein overexpression actually produce more apoptotic dopaminergic neurons, as shown here, potentially explaining why the organoids size are smaller. And we just looking at using a separate assay to look at the viability, and indeed the mutant organoids showed decreased cellular viability. To gain a global understanding of the whole transcriptome level changes in those organoids, we perform RNA sequencing. This is bulk. Now we are doing that in the single cell using single cell RNA sequencing. We see that actually quite perhaps not surprisingly that the major difference seems to be the one with the alpha-synuclein overexpression, right? That really changes the global transcriptome a lot. But compared with that, the GBA knockout did relatively much less. Fewer genes were altered, and then mostly involved actually in cell division. We do not understand why and at this moment. But when we look at the alpha-synuclein, that's where we see a lot of genes that are implicated in Parkinson's. That's showing up. So that shows us, give us some assurance that this model might be useful to understand the consequences of having extra alpha-synuclein. Okay, so at this time, all the data I showed you actually we kind of already know that through early other model systems in the mice or in the human cell lines. And we are really interested in moving forward. So we decided to see if these organoid models can really bring out something new about the human pathology. So in one of the attempts, what we did is actually we made a double mutation. So we in other words, we overexpressed alpha-synuclein in the background of GBA knockout. And here is a very basic quantification, a characterization of cell lines. The ES cells are actually fine. And we are again seeing that the G6 activities is completely abolished in the double mutant. So, but then when we look at the alpha-synuclein aggregation, that's where we see something quite striking. That we see this kind of hollow staining patterns for alpha-synuclein in this case. Ranging the size of this diameter is about between 1 to 2 microns. And this immediately raised the possibility that this could be actually Lewy body-like inclusion, because that's how the Lewy body in the human cases look like under immunostaining. To quantify that further, we used electron microscopy. So this is to show that, and the top panel is the one that we have from the human midbrain organoids. And the bottom is actually the data from the real human post-PD patient postmortem brains. So you can see that there indeed the organoids that we produce contain those kind of eosinophilic structures that are really similar to the Lewy bodies, but not quite, right? They have similarly a hollow stain positive with alpha-synuclein and a ubiquitin positive core, and they have this granular inside and the filaments outside in this kind of TM images. However, there are major differences. One, the size is very different. So as I mentioned, the size of the Lewy bodies that we can get is ranging about between 1 to 2 microns, and they are typically around 1.5. However, in the human cases, you can have up to 10, right? So they are way smaller, and the frequency that we get is actually not very high. It's quite infrequent at this stage. We also want to be sure that this is not a defect with this particular H9 line that we engineered all these mutations to. So we decided to look at the multiple line to validate. So this is really a summary slide to show you that we tried a couple of things. First, we use the H1 and we did the same approach. So we engineered the mutations, the double mutations. We also take the control iPS cells and this is a healthy donor iPS cells we got from Korea Institute, as well as patients with alpha-synuclein triple triplication, also from this Korea Institute, and we treated them with CBE. CBE is a small molecule inhibitor for this for the GCase activities, and indeed only in this case when you have iPS cells and together with the inhibitor for the GBA, then you have this Lewy body-like ring structures. We also take the other approach. We take the patient with GBA mutations. This is because the common mutations such as N37S, this is one of the lines where we had here, also L444P. Those patients carry those specific GBA mutations actually do not have a complete abolishment of the intermediate. It's a partial loss of function. But nevertheless, in those cases, if we put in alpha-synuclein, overexpressed alpha-synuclein cases, we're still able to see Lewy body-like ring structures, as shown here. And this is the right panel is a quantification. Okay. Again, looking at some of the developmental sort of stages characterization of those organoids, we pretty much see the same that the early lineage seems to be by and large unaffected, but we have a specific decrease in the proportion of dopaminergic neurons in the older age organoids. All right. Okay, so really quick summary here. I showed you that double genetic perturbation of PD-linked genes can result in de novo formation of Lewy body-like structures. I like to highlight the word de novo because there are ways to get Lewy body-like structures, but probably mostly using preformed fibrils, injections or transfections. As far as we think, this is one of the fewer models, this organoid models is one of the fewer models that shows this de novo formation of Lewy body-like structures. And therefore, we believe that this is actually an experimentally tractable system to dissect Lewy body pathology, formation, or their functional consequences, for example, and also to look at why, to have a way to dynamically monitor and study the process of dopaminergic neuron degeneration. I hope I still have a couple of minutes to share the very last bit of work. We are also, we're attacking this question from several angles. One of this is to look at what's actually different, why the double mutation bring out Lewy body structures, but the others, for example, single alpha-synuclein overexpression cannot. We notice that despite the fact that the monomer levels are similarly overexpressed, produced, but the high molecular weight species is different, right? So here, this is the lysate, the cross-linked lysate from the double mutation. You have a significantly more high molecular weight in this case. Another way of looking at it is actually using the PMCA. And again, we show that we see evidence that the double mutation produce some kind of the seed structure, the seeds that produced by the double mutations are somewhat different from the single mutations or GBA knockout only. Last, we're using oligomeric specific antibody for alpha-synuclein. And this is the dot plot, and this is the sandwich ELISA assays to show that looks like the more oligomeric alpha-synuclein strings in these double mutations as compared to the alpha-synuclein overexpression only. Okay, so I'm going to stop here. This is the acknowledgement slide for the people that have done the work. A lot of support is actually obtained by my postdoctoral mentor, Professor Tan-Yin King, who is a clinician scientist on Parkinson. And the support, the grant support is from MRC from Singapore. This is probably the most important slide. As mentioned, my lab has just started because of COVID. So I designed this cell culture 1 year ago. because there's no cell culture room for me to start my lab. So this is why only a couple of days ago now this becomes a reality. So now we're seriously about studying, and I have a small group now, and we don't only talk about research. We actually love food, and recently I got the funding support from National Research Foundation, and really looking forward to have talented postdocs and technicians and to join me to further work on this part of the work. And definitely I'm open to collaborations as well. Okay, thank you very much for your attention.
Okay, great, Alfred. It's exciting to start a new lab. So there's a couple of questions on the chat bar. So
okay.
Maybe David, do you want to ask a question yourself?
Sure. If you overexpress synuclein in the non-TH cells, positive cells in the organoids, do those cells also have some morphological or any of the changes you see? Or is it really specific to when overexpressed in just TH cells?
Good question. Thanks, David. In fact, our organoid system is it doesn't allow us the published work. We didn't use anything to restrict the overexpression of alpha-synuclein in the TH. Therefore, it's a pan overexpression, and so we definitely see evidence that other cells, non-TH positive cells, overexpress alpha-synuclein. However, when we made the analysis, we focused on TH positive cells. We didn't really look at that. It would be interesting to look at those cells as well.
I guess I'm trying to wonder why dopaminergic neurons are so sensitive. Maybe that could give a clue. Like
right. We don't know why. We see that the GABAergic populations are relatively not affected. But the dopaminergic populations have a viability issue after a while. That's what we reproducibly see. Yeah.
Okay. It's a question from Mayak. Do you want to ask a question yourself? So what's the brown thing? Is healthy brain?
Oh,
slides. Yeah.
Sure. Is she gonna? May, do you want to ask a question, or I can read out for you?
Yeah, go ahead.
Okay, so the question is, what's the brown thing on the healthy patient's brain? That's the dopa. That's, I believe you are talking about the neuromelanin, right? So that's the that's in the healthy patient's brain, healthy people's brain. But in the PD patients, you actually see depigmentation, which because of the loss of the dopaminergic neurons.
Okay. Here's a question from nobody, but you're welcome to ask yourself. Okay. So how long it takes for the Lewy-like structure to appear? How long it takes?
Yeah. We certainly do not see it up to 3 months. It has to wait a little bit longer. But actually depends on, it's quite interesting. This is depending on who culture it and how nice you are to the organoids. So I think that speaks to the experimental details a little bit. The reason why we are able to see Lewy body pathology actually quite early, right? Even 4 months, 5 months is nothing compared to the decades of the time, the natural time that takes for the Lewy body, for that to develop. It could be because the organoids are quite stressed, and we are actively looking into that. Oh. So fortuitously, in this case, stress might help us bring out this pathology. So we do see some kind of variations in terms of when you can see that, but not. I would not say that you can get that quite early. It's definitely quite a late stage phenomenon.
Okay, great. Is there any more questions?
Yeah, Alfred, I got a question. Great. That was a great job, Alfred. So I know that for lots of dopamine neurons, they'll make these huge axonal arborizations that just go all over the place. And some people think that adds that maybe there's this big metabolic stress, or there's some other demands on the cells because they have to maintain this huge compartment. Have you noticed as these 3D organoids grow, do these cells start sending an axon that just sort of weaves its way around all over the place, or do you have to give it enough time to start making a substantial axonal arbor? And do you think that matters for the kind of the ultimate metabolic stress that the cell is under?
Thank you. I think you asked a very key question. Indeed, I think all of what you mentioned is true. Let me tell you what we see in the organoids. So in one of our this is early work. So we use these wild type cells. So it's a little bit difficult to visualize them unless you section them and fix them. Of course, but so since then we made a TH reporter. And so we label specifically the dopamine neuron screen in our hands, right? So we are able actually to visualize the neural algorithms, particularly in the neuron, how it grows and extend. And we see exactly what you mentioned. So we look at the starting from early days, we can see a little bit of green, but I was very amazed by how much this arborization can grow. Indeed, in the end, we see quite a bit. But the truth is, I don't think this can, in this case, that's a limitation, right? That's where goes back to what I mentioned, all models are wrong. Here, the dopamine neurons have nowhere to go. The axons cannot project to its own cardinal partners, the dorsal striatum. So it's probably synapsing back to and making connections back into the cells among the organoids. That's for sure. So we don't see that kind of typical arborization tree that is very classic in the textbooks, that kind of things, where it can be up to like 1 meter or something. We certainly do not see that, and in a way, I think that limits the many of the phenotypes that we can deep into, because without that metabolic stress or demand from those hugely arborized axon termini, it will be actually quite challenging to make them die. so we can't. We're struggling a little bit, and we are thinking about some other ways to sort of bring that aspect out in order to study the vulnerability issue with those dopaminergic neurons.
All right, thanks, Alfred.
Thank you.
Great. Yeah, we should time the Morgan second talk, but David provide the comments. Definitely has something in my mind as well. It's like co-culture striatum. You can find the correct target. So let's go on. Okay. Anyway, it's second Alfred, the great talks. So Aaron, to Istvan.
Great. Okay. Thanks, Alfred. Really exciting new model. And now I'd like to introduce our second speaker, Dr. Istvan Katona. Dr. Katona is the Knauss Family Chair in Addiction Sciences in the Department of Psychological and Brain Sciences at Indiana University. He received his master's and PhD in Hungary. And then as a PhD student, he made important discoveries about cannabinoid receptors and endocannabinoid signaling, and then did a short postdoc in Germany before returning to Hungary, where he rose to the ranks and then became head of the department in the Institute of Experimental Medicine, Hungarian Academy of Sciences. And then very recently, he moved to Indiana University, where his lab continues to push the forefronts of using small molecule to probe fascinating aspects to cell type and subcellular compartment signaling, and invented this new technique, which he'll talk to us about. So thanks so much for joining us, Istvan.
Thanks. Thank you very much for this really prestigious invitation. Please let me know if you can see my screen.
Yep.
Do you see the full screen? Yes.
Yeah.
You should see the full screen. Okay. So this is an amazing idea to organize this NeuroZoom, and I really appreciate the work of Zilong and Aaron to put this together. And so thank you very much to invite me to present our work. And for everyone, I wish you good morning, good afternoon, good evening, good night, whatever you want. I hope I can entertain you with some beautiful slides today. And if you're not, if you are during the night, just feel free to sleep. And I have the privilege to work with amazing generation of young, motivated neuroscientists. And the project I would like to present today was started in Hungary. And then this year, our team moved to Indiana University. We got a wonderful opportunity to extend our research. So we completed this project already here in Bloomington in Indiana University. And Of course, in this community, I don't really need to introduce that this beautiful diversity of the brain. It's very exciting for us as scientists, but it also represents a major conceptual and medical challenge. So we know from functional brain imaging studies that there are more than 1,000 different regions in the brain which contribute to different aspects of our behavior. So the brain circuits in these brain regions are pretty unique in how they communicate with each other. And then this amazing revolution in single-cell RNA sequencing, as well as in many other technologies, revealed that the cell type complexity of the brain is really perplexing. There are more than 1,000 different cell types in the human brain, as it is estimated now, and altogether about 200 cell types have been cataloged in all other organs. So this really shows you the diversity. And then the vast majority of our genes are expressed in a highly cell type specific manner, both at the qualitative as well as at the quantitative levels. So altogether, this amazing complexity represents a very important challenge for the following postulation. What we propose, and of course this is a kind of common knowledge in the field, that physiological and pathophysiological plasticity processes are associated with cell type specific molecular changes that can often occur at the nanoscale level. So if you want to understand the different pathophysiological processes in certain brain disorders, then we really need to use techniques which can help us to reveal molecular changes at the nanoscale level and in a cell type and even in a subcellular compartment specific manner. So today I would like to present some introduction how we used immunostaining and antibodies for storm super resolution imaging to measure molecular changes. And then I would like to present a brand new story about how we use pharmacological tools to better quantify nanoscale molecular distributions in brain circuits. The majority of this new work has been done by an exceptionally talented young PhD student, Susanna Prokop. And of course, we collaborated with amazing chemists, Péter Ábrányi-Balog and György Keserű from Hungary. They are organic chemists and they developed several very important pharmacoprobes for us. My background is in endocannabinoid signaling. So you may know that the psychoactive compound in the cannabis plant, THC, activates a receptor which is called the CB1 cannabinoid receptor, and this receptor is activated by a retrograde messenger called 2AG, which derives from a postsynaptic enzyme called DEG-lipase alpha. So DEG-lipase alpha is cross-linked with HOMER with perisynaptic receptors such as the group 1 mGlu receptors. And then when 2AG travels backwards, it activates CB1 receptors, which can then block neurotransmitter release. And there is another enzyme, I will talk a little bit about it, it's called MGL, monoacylglycerol lipase, which inactivates 2AG signaling. And in the last 2 decades, thanks to the work of many labs, including our, now we have a consensus view about the molecular architecture of retrograde synaptic signaling mediated by endocannabinoids. And why we got really interested in the question of cell type specific nanoscale changes was a completely unexpected observation which we did in the Fragile X mouse model. As you may know, that the leading theory in this research field is that enhanced group 1 mGlu signaling is causing lots of downstream signaling changes, and that can lead to several synaptic and behavioral defects. And we know that mGluRs can trigger endocannabinoid-dependent long-term depression at synapses. So in collaboration with Olivier Manzoni and Daniele Piomelli, we measured in the mouse model endocannabinoid LTD in Y-type and knockout, and we had a very surprising observation. It was completely opposite to what we expected. Instead of an increased mGlu-mediated endocannabinoid LTD, we found a complete absence of endocannabinoid LTD in these mice. So this was very, very difficult. It was going against the dogma. So of course we wanted to figure out what can be the molecular explanation. And more than 10 years ago, we proposed that based on the perisynaptic distribution of several important molecular players, such as mGlu5, GQ11, PLC betas, and deglipase alpha, which are all cross-linked at the edge of the Homer scaffold, at the edge of the postsynaptic density in the so-called perisynaptic machinery, we wanted to use electron microscopy to figure out what's happening with these molecular players. So first, we were really disappointed because mGlu5 had an absolutely healthy perisynaptic distribution. Here you can see the immunogold particles in black, which represent the position of the metabotropic glutamate receptor 5 receptor at the edge of the synapses, and the nanoscale distribution was exactly the same. On the other hand, when we measured the nanoscale distribution of the endocannabinoid producing enzyme deglipase alpha, we had a very, very surprising observation. Although the number of the enzyme copies were the same in the dendritic spine head, we found that the majority of the enzyme cannot reach this nanoscale perisynaptic compartment and remains approximately 100 nanometer away from its final target. So for me, it was really surprising and really exciting. First of all, we only found it in excitatory synapses, but not at inhibitory synapses, so it was a cell type specific phenomenon. And it also suggested that a 100 nanometer nanoscale targeting problem can lead to a complete absence of form of synaptic plasticity, which then can contribute to several behavioral deficits as well. So how to study nanoscale molecular changes in a cell type specific manner? My favorite technology, I did my PhD mainly with electron microscopy, and the reason why I really like it, because if you use gold particles, then you can really quantify nanoscale distributions, such as in this paper when we described the presynaptic CB1 receptor, or in this paper when we described the postsynaptic deglipase alpha enzyme. And what is really beautiful in electron microscopy that you can visualize all the membranes. So for your molecular label, you can immediately provide a subcellular context, which can help you to make functional predictions. On the other hand, the major weaknesses of electron microscopy is that it's really time consuming, and it's very difficult to study several proteins at the same time. And altogether, it has a very low yield if you want to measure molecular changes in a quantitative manner and then in specific target cell types. So we were very excited when super resolution microscopy approaches came out, and especially we were really delighted about the arrival of STORM super resolution microscopy. So I don't present you all the details, but basically you have blinking fluorophores. So each fluorophore representing position of a single, let's say, cannabinoid receptor are blinking, which means that you can take like 10,000 different pictures and then you can merge them together, and then you will get the final picture in your microscope. Or if you are a French impressionist painting like Henry Edmond Cross was, if you can use a pointillistic technology, so you can put down individual patches onto the canvas and then you can have a beautiful painting you can look from a distance. So I was really delighted that this painter predicted the arrival of STORM imaging. But how does that look like in real microscopic images? So this is the CB1 staining in the hippocampus with conventional F-fluorescent microscopy. You can see the axon terminals and the axons. And there with confocal microscopy, of course, you have a much better signal to noise ratio. And on the other hand, if you zoom in and deconvolve, this is the maximum you can get with the confocal microscopy. But when you switch to STORM super resolution imaging, you can see the localization of the individual fluorophores representing your individual cannabinoid receptors. And then you can combine it, of course, with several other molecular markers, such in this case with BASUN, which is a presynaptic active zone protein. And the localization precision of your fluorophore is very similar to the localization precision you can achieve with electron microscopy. So this was very exciting for us, but as an electron microscopic, I had a major problem. With cannabinoid receptors on GABAergic terminals, there are several thousand copies, so we knew that these large axon terminals belong to GABAergic neurons. neurons. But what about these little dots? Do they belong to excitatory terminal, astrocytes, or is it just simply background? So we don't have a context for our molecular imaging. Even with bassoon, when you have the synaptic labeling, it looks like stars during the night on the sky. So we decided to build together a confocal microscope and a super resolution microscope, and also we used whole cell patch clamp recording to fill individual neurons with biocytin, and then aligned the confocal image with the storm super resolution image, and thereby we can provide the subcellular context. So one can start with a conventional region of interest. Then we have an automatic contour analysis we developed, which can figure out the important subcellular compartment in which you want to do nanoscale molecular measurements. And then you can filter out all molecular localizations not belonging to your favorite subcellular profile. So this approach helped us to do many studies. I will just show you one very brief to illustrate the logic of our approach. So first we do electrophysiological recordings from specific cell types. During the recording, we fill the neurons with biocytin via the patch electrode. Then we can do a neurolucid reconstruction, so we can tell the cell type. And then we can go to this combined confocal and storm setup, and then we can visualize the nanoscale distribution of the cannabinoid receptors on the axon terminals. And this approach helped us to figure out some very interesting cell type specific differences. And also this is really good for people who want to do behavioral pharmacology because it's really high throughput. So for example, in this experiment, we did 6 days subchronic treatment with delta-9 THC, the psychoactive compound of cannabis. And then compared to the control animals, we found a really robust downregulation of the cannabinoid receptors from the identified axon terminals of basket cells in the hippocampus. We found that there is an approximately like 85% downregulation of the receptor. Of course, it was very important to figure out that after 6 days, it is irreversible or the receptors come back. And then we found that after 6 weeks in this animal model, we have a full recovery. You can also do kind of dose response curves. So we also used a lower dose, which is approximately like in the therapeutic cannabis preparations. And we had a much milder downregulation and molecular tolerance of the cannabinoid receptors from the identified axon terminals. So all these studies have been done with antibodies, but unfortunately quantification with antibodies is not always trivial, and they have several weaknesses. And of course, there are many target proteins which are embedded into large macromolecular complexes, and basically they are not accessible for antibodies. And on the other hand, as I hope I could convince you, that with the super resolution imaging, you can really have a very nice image about the nanoscale distribution of specific proteins. And then this is really a conventional technology in life sciences for more than 40 years. On the other hand, the size of the antibody is pretty large, and therefore tissue penetration is never homogeneous. So therefore it's very difficult to quantify your target proteins, as well as there are several layers, which results in technological variability. So then we had the idea that people used autoradiography successfully for several decades, where they used small molecules like 1 nanometer sized receptor ligands. And usually you very precisely know the stoichiometry, that how many ligands should bind to a single receptor. Unfortunately, autoradiography, although it has been used for decades, mainly by actually pharmacologists and neuroanatomists, is not really used anymore that often because it has a very limited spatial resolution. So we saw that why not to combine the advantages of these two technologies, and why not to use small molecules tagged with fluorescent dyes amenable for super resolution imaging to combine the strengths of the two technologies. So we very recently started to develop these small molecules, which are becoming much easier because the crystal structure of target proteins are becoming really readily available. And then one can play around with the help of a chemist to figure out which kind of ligands would be the useful with in silico molecular dynamics stimulations. And then you can figure out the linker lengths to really keep the fluorescent molecule at the right position together with your receptor agonist. And then you need to do, of course, the conventional pharmacological characterization, and then you can test in a hexel preparation how good your fluorescent pharmacoprobe is. So in this case, we developed the fluorescent cannabinoid receptor ligand, the fluocannabinoid, and we got a very nice, beautiful, specific labeling. And then we went to this super resolution microscope, and then we started to correlate and combine pharmacostorm and immunostaining against the storm. And we did displacement experiments to prove the specificity of the labeling. And you can see that using a CB1 receptor antagonist treatment, we could completely eliminate the pharmacological labeling on the plasma membranes, but we still had the same immunostaining-based immunostorm labeling. So then, if it works well for G protein coupled receptors, why not to try other other major protein families? For us, the MGL enzyme is a very important enzyme because it is degrading the synaptic endocannabinoid 2-AG. So in collaboration in the Netherlands with the group of Mario van der Stelt, we used an inhibitor of this important enzyme, and using the same logic of displacement analysis, we could provide evidence that for enzymes one can also do nanoscale pharmacological measurements. Then another group we wanted to target were the ion channels. Usually it's very difficult to develop antibodies for ion channels. And we used in this case bungarotoxin labeled with far red dye, good for storm imaging, and again an antagonist of the alpha-7 nicotinic acetylcholine receptor could completely eliminate the pharmacological staining measured with storm imaging. So after demonstrating the broad applicability of this approach, the final next important question was as whether we could study also available medicines, molecules, which we don't really know too much about. And we chose cariprazine, which is a pretty new, very recently approved medicine. It was developed primarily against schizophrenia spectrum disorder, but now it is accepted for all forms of bipolar depression, and also it is available now as add-on therapy in as a major depressive disorders. And more than 500,000 patients have already received cariprazine. So cariprazine is not really known how it helps the patients. What is the neurobiological explanation for that? What we know that it prefers dopamine 3 receptors very much compared to D2 or serotonin or other receptors. On the other hand, these through dopamine receptors are pretty much like a little brother compared to D1 and D2 dopamine receptors, although there are really beautiful, exciting new papers coming out, but we still know much less about these receptors. And importantly, currently there are no specific antibodies available against these 3 dopamine receptors. So we tested the idea in the final set of experiments if we can use a fluorescent version of cariprazine to target these 3 dopamine receptors in the brain. Again, we started with hexel experiments to do some basic pharmacological treatments. The dopamine 3 receptor antagonist could completely eliminate the pharmacological labeling on the plasma membrane of the hexels. And then we did some quantification to characterize this molecule. And I was really excited to see that basically you can get back this pharmacological textbook sigmoid shape curve about the binding profiles of the fluorescent cariprazine. And storm microscopy is a type of single molecule localization microscopy. So it has an amazing detection sensitivity for single molecules. So therefore, even in very low concentrations, even at subnanomolar concentrations, we could measure specific binding of cariprazine. So in this case, when we did this experiment, this was basically binding of very few copies of fluorescent cariprazine to the plasma membrane. So if it's useful for pharmacology in cell culture preparations, of course, as a neuroscientist, for us, it was very exciting to figure out what is visualized in the brain. And one reason why not too many people are working with these three dopamine receptors, because they are much rarer compared to D1 and D2 dopamine receptors. And when we did the first stainings, we struggled a lot with fluorescent cariprazine. And as you can see in this forebrain hole image, there is not really a labeling. However, Susanna Prokop had such good eyes that she picked up some little yellow binding at the ventral surface of the brain in a region which is called the islands of Calleja. And then we did, of course, high magnification imaging, and we found that the fluorescent cariprazine was binding to the so-called hilar subregion of the islands of Calleja, not really to the granular subregion. And it was indeed D3 dopamine receptor dependent because it disappeared from the D3 knockout mice. And this is very, very exciting because this region in general gets very strong dopaminergic innervation. On the other hand, we didn't really see the binding in nearby regions, but also in the hilar subregion, we could see a specific dopaminergic innervation. So altogether, this suggested that dopaminergic innervation and D3 dopamine receptors in the islands of Calleja may be affected by cariprazine treatment even in patients. So to kind of provide an in vivo evidence for this view, we used unlabeled cariprazine treatment in live animals. And when we treated the mice with vehicle, we could see a beautiful fluorescent cariprazine staining in the islands of Calleja. On the other hand, the unlabeled cariprazine could fully displace the binding of the fluorescent cariprazine. So the cariprazine which was injected in vivo into these mice were displacing the fluorescent label we added later on. There was a kind of view in the literature that dopaminergic terminals may have D3 autoreceptors. So first we started measuring TH containing dopaminergic axon terminals. Interestingly, in this in vivo experiment, we did not see colocalization between the dopaminergic terminal and fluorescent cariprazine binding on D3 receptors. So then we started to do more anatomical work on the islands of Calleja neurons, because these neurons are really understudied, unfortunately, in the field. So we again used patch clamp electrophysiology, and we filled the neurons with biocytin, and we found that there is always a specific hilar process of these neurons, and there are little thicker so-called granular processes of these neurons. And then we developed the biocytin, and then we did electron microscopy to provide evidence that the hilar process forms synapses on postsynaptic profile. So this is the axon of the cells, and the granular process receives afferent synapses. So you can see the yellow fluorescent cariprazine binding is always in the region where the axons are located. And it suggests that cariprazine will bind to the axons of these cells. But in the final experiments, we wanted to provide a direct evidence for that. So again, we combined the fluorescent measure, the physiological measurements, and the anatomical reconstruction, and then measurements about the axon terminals and the axons of the islands of Calleja granule cells. And here in this image, which is my favorite, you can see the individual fluorescent cariprazine molecules are binding on the surface of these axon terminals. And altogether, this suggests that one major action of cariprazine may be is that it can control the output of the islands of Calleja granule cells and may control the dopaminergic volume transmission, because we never really saw a kind of synaptic-like clustering of these receptors. And why this is very, very important, because I think this also highlights that there is an understood brain region, which is called the islands of Calleja. When we did three-dimensional reconstruction, it turned out that the cariprazine binding with the axons actually links together all the cell masses, and then we did the 3D reconstruction, and we got a pretty surprisingly large size for the islands of Calleja. It basically reached about 1/3 of the nucleus accumbens size. So altogether, I would like to highlight that this brain region is really worth for future exploration for all of you. And I was amazingly delighted to find a beautiful paper from the lab of Ming-Hong Ma from UPenn, which came out just a week ago, showing that this brain region has a critical role in grooming behavior in mice. And Vraylar is the latest blockbuster drug in psychiatry. It became blockbuster last year. It is already advertised at many places, even during the election night. And I hope that the Pharmacostorm work and the research can contribute to a better understanding of its beneficial or perhaps potential adverse effects. The final take-home messages for today are that the STORM super resolution imaging, both the immunostaining based and now with the pharmacoprobe based approaches, are really useful for measurement of nanoscale molecular distributions and their changes in a cell type specific manner in complex brain circuits. And this novel antipsychotic cariprazine preferentially binds to D3 dopamine receptors on the axons of islands of Calleja granule cells. I also appreciate the help of several grant agencies who supported our work. And this picture was literally taken from our postdoc office. This is how we celebrated the acceptance of our Nature Communications paper. Thank you very much for your attention, and I hope you have a few questions.
Thanks so much, Istvan, for that great talk. We're now open for questions. Ming Hong, do you want to ask your question?
Hi, nice talk. So I just wonder whether you see that binding to hippocampal D3 receptor in addition to the islands of Calleja.
So I must admit, this is the project we are working on to figure out even lower copy number binding sites for D3 using this amazingly sensitive tool. Already in the first paper, we compared the binding with the nucleus accumbens because there is a population of D3 containing neurons there. And we could show that, that in the D3 knockout, fluorescent cariprazine binding in the nucleus accumbens disappears, but the binding density is at least 10 times lower, even much lower than 10 times lower. So of course, this is now a very important question to try to figure out which cell types and which subcellular compartments contain this receptor and the binding density, and how that binding density changes in association with different brain disorders. Addiction is an important focus for our team.
Okay, other questions?
Okay, hello, can you hear me? I have a question.
Yes. Hi, Jim.
Okay. Oh, hi, Carol. Hi, sorry. Who see something? Oh, hey, Katona. So I have a question. So you did the STORM to imaging the CB1 receptor in the buttons, right? So after you treat with THC, you got you got a dramatic decrease of CB1 receptor. So what happens to this receptor? Did the degradation or just go somewhere else? Have you checked the consequence of the distribution of this receptor after treatment?
So after 6 days, this is actually this is a general essay in the field for cannabinoid researchers. For many decades, so we used exactly the same assay, and we found that approximately 75% of the receptor disappeared for immunostaining, so we couldn't visualize them. Perhaps these proteins were degraded, and we found an increased internalization, which was about 10%. Was it this question? Was it your question?
Yeah. It's this question. So they are internalized, but so they are internalized. Did they go to the somat or go to somewhere else, or but basically?
We don't know that. We only measure the axon terminals. So.
Okay, yeah. Basically, in my my knowledge, I think the GPCR after internalization, they will recycle back to the memory to.
Yes,
actually,
With Rohini Kunar, we have a paper like 15 years ago, and Mark von Zastrow's lab also showed that there is an equilibrium regarding downregulation via lysosomes in the axon terminal, or there is a recycling, and it's always the concentration of the receptor agonist which determines how this equilibrium is shifted to the downregulation of the proteins. Of course, when you have a high dose and chronic treatment, then the lysosomal degradation comes into picture. But if you have a low dose, then the system is usually recycling the majority of GPCRs, of course.
Okay, yeah. Thank you so much. This was a nice talk. Thank you.
Thanks for your question.
Okay, other questions for Istvan? Okay, thanks so much.
Great.
It's long. Any questions? Okay, no.
Great. Thanks.
Yeah, great. Thanks for sharing that exciting new technology with us. It's fun, and thanks
very much,
Alfred, for coming as well. See everyone next week.
See you. Bye bye.
Okay, bye.